Assalamualaikum. Dr. Afi tak dengar? Not very clear. Okey dikit. Okey sikit.
Ya, kurang jelas Dr. Alfi.
Kurang jelas.
Macam mana sekarang? Okey. Mendengar? Halo.
Salam, Doctor. We do not hear very well.
Ya, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Before we start our presentation today,
I'm sorry, Dr. Afi. That's not very clear. Not clear. I think because the system is not proper, yeah.
how is it now? Can you hear? Dr. Mohammed, can you hear me? Dr. Mohammed?
Yes, can, but not very clear, but it's okay.
Yes. So can I continue, Dr. Mohammed? Can you hear me?
Yes, Dr. Afi.
Yeah. Okay. Yeah. So we are now going to proceed to the top Tarik topic. So here, so it is the platform for us to know further regarding our kuliah. So then further that we can collaborate with other. Okay. For today, we have two speakers from our kuliah. First is the assistant professor Dr. Mohammed Surana. Then after that, assistant professor Dr. Thomas. So Dr. Mohammed is from the Department of Psychology. For the Dr. Thomas from the Department. The first speaker presents this experience entitled "Phase 1 Microencapsulation for the Tarik particle." So we will present within 1 hour, including the Q&A. Okay, without further ado, I'd like to invite Dr. Mohammed to that research. And now the process you can share your slide.
Assalamu alaikum wa rahmatullah. Can I start, Dr. Alfi? Uh,
Okay, before that, we would like to announce that we have real Tarik now, that has actually Tarik. So anyone who would like to take their Tarik, you can come to this meeting room or KOP meeting room for first class. Okay, continue, Dr. Rahmat.
okay. You can see my screen.
Yes, yes.
Cannot tell.
Okay. Bismillah ar-Rahman ar-Rahim. Salatu wassalamu ala Sayyidina Muhammad wa ala alihi wa sahbihi sallim. Thank you, Dr. Alfi and School of Pharmacy for inviting me to this Tarik session. So today I would like to share some of the theory and then our work that is related to nanoparticle preparation using a specific technique, which is the electrospray. So our title here is electrospray as a one of microencapsulation technique for tailored size particles. So it's not only for nanoparticles, but for all sizes range from millimeter scale to nanometer scale. First of all, let's explain what is the principle of this electrospray technology. Because many of the researchers try to prepare nanoparticles using the conventional methods that are usually like manual method, and commonly they do not require specific equipment. They are just need stirrer or homogenizer or ultrasonic, basically just for making the emulsion or for like mixing. But with a little bit more equipment or a little bit higher level equipment, we can actually prepare nanoparticles or microparticles or beads, millimeter scale particles in much more advanced technology. So what is electrospray? Electrospray, or what is called sometimes electrohydrodynamic atomization or electrohydrodynamic spraying, is a method for liquid atomization. So simply it's a liquid atomization technology like this one, which is like very common now, this atomizer that used for sanitization. This electrospray is basically, in the principle, it's similar. It's an atomizing technology. It converts a bulk of liquid into spray, but not using the pressure or the mechanical push, something like this, but it uses electrical force. So the spray happened because of the presence of high voltage power supply. This power supply provides the electrical field between the nozzle spray and the receiving electrode or the receiving surface or receiving liquid. So you will get a nice spray of droplets. So these droplets later can be converted into particles using different mechanisms. So, but the principle of electrospray is it's basically a spraying or atomizing technology. It's atomizing technology. So here how it appear actually. This is from not our work, but it's from YouTube, because when it comes to nanoparticle atomization, it become very difficult to take the photos. Okay, so this is basically the principle and how it looks like when it's spray for nanoparticles. But basically, the size of spray droplets range from millimeter to hundreds of micrometers and can go down to several tens of nanometers. The size distribution is nearly monodisperse, and this is what is very unique about this technology, other than the spraying that have been in using the air pressure, like in spray drying. They have high air pressure that push the spray from the nozzle to get particles, which usually in micrometer scale. This spray can go to nanometer scale, and the size distribution is almost monodisperse, meaning that you will get particles almost of the same size. And the charged particles are self-dispersed because they repel each other, so they will disperse very nicely, which can avoid the coagulation. So this is also very important, and what distinguish this technology from other spraying or atomizing technologies. So now, how we can use this technology to make nanoparticles or microparticles? If we go back to the basics of nanoparticle formation, basically we can divide them into two main categories or two main principles. One principles, we call it bottom up, meaning that we have atoms or ions or molecules, a very, very tiny, like say particles or atoms, whatever. They accumulate with each other to make a nanoparticle. So meaning that we make the nanoparticles starting from its basic constituents, which are the atoms and molecules. This is basically like when you have precipitation. We start with molecules or atoms or ions. They accumulate with each other and precipitate to form nanoparticles. But the other technology or mechanism, which is more common, is the what we call up-down or something like this. We basically start with bulk liquid or bulk precursor. Then we need to cut it into pieces to make nanoparticles. So we have different mechanisms to make these nanoparticles. If it's a solid, usually by nanomil, but it's very specific in or very limited in application. If you want to encapsulate something, usually it's not suitable. Most of these bulk precursors start with a liquid that need to be separated into small droplets. Then these droplets become nanoparticles. So either the liquid is a solution that you spray, then the solution will evaporate, leaving whatever dissolved in the solution as nanoparticle. Or commonly also you can make it as an emulsion. Then you spray the emulsion to make a nanoparticle, or you evaporate the solvent to make nanoparticles, which is these are the common methods to make nanoparticles using the second mechanism. So like solvent evaporation method, we usually common polymers, we use PLGA or polylactic glycolic acid, polylactic acid, polycaprolactone. These are some of the common polymers used. We have also ion gelation or conservation. Some of the common polymers is sodium alginate or ketosane or albumin. In this method, the droplets that we spray, we receive them in a solution that cross-link them. For sodium, for example, if you spray alginate solution to calcium chloride solution, it will cross-link, the calcium will cross-link alginate to convert the droplet into particle. So it depends on the size of the droplet, you will get a different size of the nanoparticles. Similar thing also for ketosane, also for albumin, we have can be coagulation or conservation. Sometimes we don't need to cross-link the particle, but simply evaporate the droplet on the way, like while the particle is being sprayed, we simply evaporate, let the solvent evaporate on the way. It will make a nanoparticle. Okay, if you didn't hear about electrospray, I'll tell you two very related technologies that are related to electrospray. First one is electrospinning. Electrospinning is considered like the brother of, or the big brother of electrospray, because electrospinning is quite more common. It's used to make nanofibers, like the expensive face mask that contain these nanofibers. This is using the same, basically, it's the same principle, the same technology, but we spray highly viscous solution. So when it's spray, it make fibers on the way. So basically, it's the setup and the materials almost same. The other very related technology is called also electrospray, but it's not used for nanoparticle production, but it's used for in mass spectrophotometry. If you wonder how mass LCMS, sorry, LCMS, how LCMS work, you start with your mobile phase, then electrospray basically spray your mobile phase that cause evaporation of the solvent and keeping your atoms or molecules that go to the mass spectrophotometer detector. So basically, the one that spray your mobile phase in LCMS is actually a electrospray setup. It looks exactly like this, but it's used for that is the most common application for electrospray. The other application is using using it to prepare nanoparticles or some kind of coating like here. But this is actually some of the related technologies that are quite common, but you may not know that it's actually related to electrospray. So these are some of the atomizing devices or technologies. There are many using different techniques, like the most common one, actually the pneumatic, which using the air pressure, commonly used in spray drying. And we have the using electrical field in electrospray. And the third also common one is the using acoustic atomizer or ultrasonic atomizer, which is now is common in your like perfume atomizer, the one that you put liquid and some perfume and start to giving this vapor, but it's not actually by heating. This vapor is called vapor. It's come from atomizing device using ultrasonic. These are actually the most common three atomizing technologies used or related to nanoparticle and particle production. So the electrospray has the following advantages over conventional mechanical atomizers or other droplet generating methods. So why we focus on droplet generating methods? Because as I mentioned, we need to cut up the solution into droplet first in order to convert it to particle. So droplet size is smaller, and the size distribution is almost monodisperse. The charged particles are self-dispersed, so they repel each other. So this will avoid the coagulation. Then the motion of charged particle can be controlled, like we have seen in the video. You can put rings to control the size, the diameter of the spray, and the deposition efficiency of charged spray on an object is higher than of uncharged droplet. So basically, the charged objects are actually some kind of attracted to the surface you are spraying on, not like using like air sprayer where the particles go everywhere. Here, the particles somehow they come to the depositing surface. Actually, also similar technique for electrospray, but it's not spraying solution. It uses also to spray metal. Is the one that used for coating your sample when you do scanning electron microscopy. If you wonder how the gold transfer from up to your surface, to your sample, basically it's using high electric field, using the same technology using electrospray, but it spray the atoms of the gold, which will deposit on your sample and do this coating in nano diameter coating. So how's the droplet formation in electrospray? Basically, a string electric field is built up at the capillary outlet. So this is the capillary. and the liquid flow from the capillary using usually a pump. And then when we talk about nanoparticle, usually we need to use a high accuracy pump, like a syringe pump, similar to the one used in infusion in the hospital. So this solution, if you leave it without interacting with the electrical field or whatever, basically it will build up to make large particle like here. Then this large particle will drop based on the what we call its weight. So when its weight become larger than the surface tension that hold it up, it will fall down. But when we have electrical field applied, the particle becomes smaller, and there is some kind of force that pull it down. So by this way, the particle size becomes smaller, and even can go smaller than the size of the capillary. You can see here I'm applying a stronger electrical field or high voltage. Then you can see the particle start to fall smaller and smaller, and finally become very fine particles, which usually in this one we are in the size of micrometer. So there is two ways or two modes for spray drying. First mode is the dripping mode, and second mode is the jet mode. So basically they are, first one is the dripping mode. From the name you can see it's dripping a whole piece of the liquid to make a particle or to make a droplet. So this is a normal dripping mode, usually in millimeter scale. But if you apply higher voltage, you will get a micro dripping to make it in micrometer scale. This is one of the dripping method. We sync it with, we control it with the voice. This is a micro dripping mode we have done in our lab. And the second mode is the jet mode. In the jet mode, the particle do not drop by using that force, but instead they spray. So after building up this what we call conjet, the particles start to flow, and the liquid start to flow. Then it will be separated into particles because the liquid repel each other because of the high electrical field, and start to spray. So it will slowly go down and start to spray. And because of the charged particles, they repel each other and make that cone. So this is, we call it conjet mode. So it's very difficult to see this spray using normal photography, like normal camera. So here there is one, but it's very difficult to see. So this mode usually used to prepare nanoparticles. The previous one used for millimeter scale, micrometer scale, and this one is nanometer scale. So basically, the good thing about this technology that the same setup used for different size of particles. It's a bit different from other technologies used to prepare particles in general, because in other technologies like solvent evaporation or solvent diffusion or whatever, usually when you want to change the size largely, or you want to go from millimeter or to nanometer or to micrometer, you need to change the formulation. You need to change the solvent. You may need to increase or decrease the polymer concentration, adding a surfactant or so on to get the size you want. But in this technology, using the same formulation, we only play with the process. So meaning that we don't need to change the formulation, we just change the process. Usually there's two main variables that we need to change: the flow rate of the liquid and the high voltage value. So when we are talking about millimeter scale, usually we work about in the flow rate in mil per minute. But when we are talking about micro dripping, usually about in mil per hour, like 1-2 mil per hour, or let's say up to 5 mil per hour. But when we are talking about the nanoparticle production, we are talking about microliters per hour. usually below 1,000, like below 1 mil per hour. So meaning that you can figure out that this technology, one of the disadvantage is that it's low output, because the flow rate that need to use is very small when it goes to nanometer scale. It's just like if you want to prepare 1 mil of the liquid to make it nanoparticles, and your flow rate is about 0.2 mil per hour, you need 5 hours to separate 1 mil only. So the droplet size can be decreased usually by decreasing flow rate, conductivity, and surface tension. So basically, we don't play much with the conductivity and surface tension, because we just fix it in the formulation, but we play with the flow rate. The size of capillary also has effect, but usually is some kind of after primary experiments we fix the capillary. The capillary here we use basically syringe. Sorry, the needle similar to the one used in the syringe, but it's a blunt one, not sharp. In general, changing liquid physical properties independently is not easy task. This is why we don't change the formulation. If you want to change the conductivity, you need to add one material. This material will affect the surface tension and may affect the viscosity. So it's very difficult to change the physical properties independently. So basically, they will change altogether. So I'm always trying to avoid playing with the formulation. After we fix formulation, we play with the process. So what is the applications of electrospray in nanotechnology? It's not used only for nanoparticle production, but also for the first application is basically for micro and nano thin film deposition, which is used in pharmaceutical technology. It's used to prepare like fast dissolving film that you just put the film in your mouth and it's very quickly dissolved. Usually it's using this thin film deposition. So here the electrospray deposition is a process in which the droplets produced for electrospray deposited on a target substrate. Like you have here, let's say, aluminum foil or a metal material, because we need to use usually a conductive material. And here is the needle or the capillary. So between them, we apply high voltage power supply, and we flow the liquid from here. Then it will spray. Then usually this one, like you dissolve your polymer in an organic solvent that is quickly evaporate, for example, dichloromethane. Then after it's spray, it will make this kind of film, because the dichloromethane is very fast to evaporate, and the spray is very, the flow rate is very small. Basically, just during the flow, it will evaporate. Or even if there is a little bit, it will evaporate here. And if you need assistance, sometimes you can apply heating to quickly evaporate the solvent. So there's many applications for this one. The most common one is to prepare fast dissolving films in pharmaceutical applications. So you can get film thinner than 100 nanometer. So you really can go for very, very thin film. And it's a very controlled spraying. Like it's also can use it for some kind of coating, but it's a really very controlled, and you can go for very, very thin coating. The other technology is the particle production, micro and nanoparticle, and then millimeter, even millimeter, we call it bits. So for particles smaller than or down to 10 nanometer, sorry, so we can produce particles of small size, can go like up to 10 nanometer, and with high monodispersity. The conjugate mode operates only in limited voltage and flow rate. So during the development of the formulation and the process, we need to find out the best combination of flow rate and high voltage that get a stable conjugate. So basically, we need to get a stable conjugate to get a good and reproducible nanoparticles. Any change in liquid physical properties like temperature will change the mode. And the size of the droplets is controlled basically by the high voltage and the flow rate of the liquid. the shortcoming, as we mentioned before, it's a low throughput because of the slow process. Rapid mass production can be used, can be done by simultaneously operated capillary nozzles like the one that, like in this setup, and the one that we see in the first video, where you can operate several nozzles together. So using the same high voltage and the same pump, but you design the array of emitters to make to increase the throwout of the spray. So this is basically when are talking about organic solvent, the technology used to make particles basically is by evaporation. So you spray the particles on the way they will basically disperse because of the electrical repulsion. Then the solvent will evaporate and you will receive particles deposited on the, for example, aluminum profile, or sorry, aluminum foil. Then you can scrap them. You will get a ready nanoparticles in a solid state, not like in other technologies where you will get the particles in a solution or the suspension and you need to centrifuge and separate them. This one needs a lot of optimization to get particles that are not sticky to each other, like well-dispersed particles. If you didn't get dispersed particle, basically you will get a thin film here. So you just need to play with the parameters to convert between nanofiber, thin film, and particles. The other application is the ketro spraying or ketro spray forming and direct writing. This technology is very similar to the printing, the inkjet printing, where we throw out particles in a specific pattern, which move using XY platform. Commonly, we convert 3D printers into this electrospray farming or writing. I work with this one. It's quite, you can make it straightforward if you convert the open source 3D printers. You can actually combine it with the electrospray to spray the particles in a specific pattern. So you can use it if you want to, like if you are encapsulating cells or bacteria or whatever, you want to deposit them in a specific pattern, you can use this one. The similar technology is the inkjet printing. If you know that some of the researchers converted inkjet printing, like our HP printer, Canon printer, they converted it into printing cells. They encapsulated cells or printed cells using inkjet printing. But in inkjet printing, the size of droplet is limited to the size of the head of the nozzle in the printer. But in electrospray, you can go smaller than the size of the capillary. So by this way, if you are using a high viscosity solution, you cannot use inkjet printing because it will block the printer head. But for electrospray, you can use coarse capillary because it will not block it will not be blocked by the high viscosity of the liquid, but yet you can get smaller droplets because of the high voltage you apply. This is one of the, not my work, but from the internet. This is the conjet, but this is not stable conjet until you get one conjet. Usually it's a stable one. So you can they use 3D printer to deposit in a specific pattern. Here is basically just scanning, but you can use whatever pattern you draw, it will follow. Now, I would like to move to some selected applications from our work in here in Korea Pharmacy, our lab. The first application is the encapsulation of active ingredients in milli and micro scale particles for oral delivery. In this one, we encapsulated the Nigella sativa oil or black seed oil, Habbat Sauda oil in alginate bits. This one in millimeter scale, but in other application, we also encapsulated in micrometer scale. So by here, we electrospray a nano emulsion of the oil in alginate into calcium chloride solution. So basically, we put in the syringe nano emulsion that we prepare using this black seed oil in sodium alginate solution. Then after getting this nano emulsion, we electrospray it into calcium chloride solution. Calcium will cross-link the droplets of alginate, converting them instantly into particles. The size can be controlled to fit the suitable application. For example, for children formulation, we need a particle size below 2.5 millimeter and also prefer to be smaller. Then we can actually control the size using the flow and voltage. We don't need to change the formulation. So this is usually we use factorial design to optimize or to find the suitable combination of voltage and flow rate. You can see when we increase the voltage from 4,000 or from 0 to 4,000 to 6,000, the size go down from 1.4 millimeter to 0.8 or 0.7 millimeter. The flow rate between 0.1 to 0.8 or up to 1 mil per minute has almost no effect on the size. But if you go higher, usually the size become bigger. So, but both flow rate and high voltage didn't affect the encapsulation efficiency. And this is quite good because we can see that the process didn't affect the encapsulation efficiency as far we are using the same formulation. This is the particles that we produce. And actually, this is the same particles that we scale up in KOP. We scale up using, we built a complete production line for these particles. But in the production line, we have not used the voltage. We just use the normal spray. But it's actually ready to use the high voltage, but that the size was suitable for our application without the high voltage. We just didn't use. So when we scale up the process, we need to find the suitable parameters that make the process reproducible and suitable for this scale up. And many times the process is suitable for lab scale, but when you scale up, it's not suitable because in lab scale, the variability is like for example, the flow rate or the distance used is quite fixed. But when you scale it up, there will be a lot of challenges. So we try to find a combination of flow rate and the distance between the nozzle and the surface of the receiving solution that are not having effect on the bit size. So in this case, to make the process reproducible and suitable for scale up, actually, we are looking for the range of flow rate and distance that has no effect, no significant effect on the size. And we commonly use this factorial design to find the equation and the effect of flow rate and high voltage. You can see here the high voltage has the most effect on the size compared to flow rate has effect, but it's not as strong as the high voltage. And also when it comes to a specific range, the effect become still within a specific acceptance limit. So the ability to electrically control the size open the door for automation. So as far we don't need to change the formulation, we only need to change the process. And this process actually is changing the high voltage and the flow rate can be done electronically. We don't need to do it manually. So by this way, we can have we can make automation of the production line. This is basically my PRGS, which I already almost finished. So we built up the prototype of the production line that use all these pumps and control the flow of the liquid. And here we control the high voltage. And we measure automatically, we measure the size of the particles using like our own setup for some simple microscope, and together with coding of the size using Python. So after we get the size, after we get the size here, the size will be transferred to the microcontroller of the production line, and it will take decision to increase the flow rate or decrease the flow rate, or increase the high voltage or decrease the high voltage based on the size that we get. So it can do any correction of the size. So it's what called PAT, Process Analytical Technology. So can automatically take picture every, let's say, 1 second, or every 30 seconds, take picture, take the size, and send the data to the microcontroller. That one was for millimeter and micrometer scale, but we also, and it was encapsulating a hydrophobic material, which is black seed oil, which can be used, similar methods can be used for encapsulating any other hydrophobic drug. Basically, since we are using alginate, so we need to dissolve it in a solvent or in an oil to make emulsion, then we spray it. But in another application, we encapsulated a hydrophilic material, which is DNA or plasmid DNA for gene therapy and gene editing. So the challenge with the hydrophilic materials, they are easy to dissolve in alginate, no problem, but the problem that they will leak out when you cross-link alginate and calcium chloride. Alginate has very big or very large pores, and the material easily can escape out from the particle. So you will get low encapsulation efficiency. So it took quite time to find the suitable parameters that can get the plasmid DNA encapsulated inside the particles. And when we are talking about nanoparticles, the surface area is very small. Sorry, the surface area is very large and can escape very easily. So we encapsulate this plasmid DNA in quite high efficiency. So this is a similar setup, basically the same setup. And we spray the particles. Quite difficult to see, but you can see the spray here. So we also come up with several studies to get the stable conjugate. So this is like multi-jet. It's not stable conjugate. Need to change some other parameters to get the stable spray, like here. So we have studied different combination concentration of the polymer, adding surfactant, and we study all these parameters of the solution in terms of like conductivity, viscosity, surface tension, density, pH, until we get the suitable conjugate, stable conjugate that produce stable particles and reproducible particles. The good thing that here is the reproducibility. For whenever you apply the same, you prepare the same solution, you put the same setup, you will end up almost with the same size, the same parameters of the particles. Not like the manual method and using like solvent evaporation. I have difficulty in solvent evaporation. Like the variability is quite high. If you prepare it today and the next month or another one, come to prepare or try to prepare them using the same method of his friend, he will get different size and different parameters. But for El Ketro spray, if you follow the same procedure, almost you will get the same particles. So again, you will always find a study for flow rate and high voltage, and commonly to find high voltage has the most significant effect on the size. So the size, when increase the voltage here, you can see the voltage is higher, is about 10 or between 9 to 12.5 or 13 kilovolt. So the size drop from 700 nanometer go down to about 250 nanometer. And this is under the TEM. These particles we apply into the cell culture and to study the cell uptake, meaning the ability of particles to go inside the cell. So we can find these particles. The particles are labeled with green, and the nucleus is blue. You can see the particles are deposited around the nucleus. They enter the cell. So we study the time-based cell uptake. And more important is to see the gene expression of the gene that you put in the plasmid. Here we put actually Cas9. With Cas9 is the enzyme used for gene editing that can cut DNA at a specific location using the CRISPR technology. So in this case, you can see at day 2 a small red color, which is the labeled Cas9 with the red fluorescent protein. But at day 7, the gene expression become larger, and you can see a lot of red color around the nucleus. And using alginate only, the cell uptake or the gene expression was not very high. So we went to do some coating of the particles. The coating was quite simple by adding the coating material into the calcium chloride solution. We used different coating materials. We have tried ketosane and polyethylene glycol and Arabic gum, acacia. So you can see in some images the coating is quite clear under the TEM, like here. And when we study the gene expression, you can see that ketosane was really having a much higher gene expression than other coating materials. And the most important part is the gene editing ability, meaning that the ability to edit the DNA and cut the DNA at a specific location. We also we found that ketosane have the highest gene editing efficiency. It's about 18%. So compared to alginate alone was almost 4% only. So you can find out how coating affect the performance of the particles. So in the previous applications, we can find that we encapsulated hydrophobic material. We encapsulated hydrophilic material, macromolecule like DNA. Another application, actually, we also encapsulated protein, but have not been published. In most recent application, we encapsulated cells in these alginate particles. We use both with high voltage and without high voltage. Without high voltage, it's difficult to control the size of the particles, but we use it for another application, which is cell preservation and cell shipping. Using high voltage, you can encapsulate the cell in the particles at controlled size, which can give you a reproducible size, because the importance of the size for encapsulating the cell here, this is used as a 3D cell culture model. 3D cell culture model, the size is important because the bigger the size, will be deeper the cells inside. So the nutrients have longer, and they are far from the nutrients from the solution. So it may get hypoxia inside. But using the controlled size, you will get like a small size that is based on the application, and the size for each particle is similar to other. So there's no variability in the size of the particles itself. So this is used for 3D cell culture. Actually, we use it to study the toxicity of some anti-cancer drug and compare it with the normal 2D cell culture. Basically, quite simple to apply. And anyone basically, it's easy to upgrade the normal 2D cell culture to 3D cell culture. So we encapsulated here 3 cell lines, A549, FG2, and U2OS. In this application, it was not for 3D cell culture itself, but it was actually to preserve the cells during the COVID-19. If you remember basically it was triggered by the, when we have this lockdown, the last lockdown actually, the cell, we have very limited entry into the incubators. So every student has very limited time to keep the cells and take care of the cells. So my student here, Batul, did this encapsulation. So in this case, the cells doesn't need much of taking care. So up to 4 weeks, basically we change the media only 1 time per week. one time per week. We change the media until the lockdown finish, or after 4 to 5 weeks, we were able to return to recover the cells back and continue to the cell culture as usual. You don't need to cryopreserve the cells here. And in another application, we use it for cell shipping. So we ship the cells using the cells were encapsulated in these alginate particles, and we ship the cells using the normal shipping. Actually, we use GNT, the one that in the Ramakutta. We use it without any instructions, without any other like arrangement. We ship the cells to Kuala Terengganu and return them back. It took about 5 days. And we have assembled, using Arduino, we assembled the data logger to monitor the temperature, humidity, and the vibration. So you can see the temperature was quite okay, 30 throughout the 5 days. And in some short time, it spiked to 40, which I expect the time it was used for delivery in Kuala Terengganu, because sometimes they use motorcycles for delivery and non-conditioned cars or something like this. The same like for relative humidity is quite about 60 to 65 all over the time. And the vibration was quite stable, but if you see the same time temperature increase, the vibration also increase, which maybe mostly correspond to the time of the last delivery. So you can find here the cells were actually alive up to 4. The first one that used for preserving the cell, there were the viability is quite high for 4 weeks. Actually, the cell continue to grow, and can see the viability, soil viability become more compared to 2D cells. This one we stop at 1 week because the cells start to overgrow and start to multi-layer grow. So we stop after 7 days, cannot proceed. We need to do the maintenance of the cell and change the media and trypsinization and so on. But for 3D cell culture using alginate, we were no need to change the media only 1 time per week. And in fact, in another extreme experiment, but it was done only 1 time, we didn't change the media at all for 5 weeks. We just keep the cell in the media for 5 weeks, and we were able to recover the cell again from the 3D and convert to 2D and continue cell culture as usual. We don't need to change even for you can keep the cell for 1 month if you don't want to take care much of the cell. This one we actually file a trademark for to make a kit for this cell preservation and for generally used for 3D cell culture, for cell preservation, and for cell shipping. And as I mentioned here, we didn't use high voltage. We just use a dripping mode, but without high voltage. But another application was not yet published. We use high voltage, and we study the effect of this high voltage on the size of the bits. And also we study we have done after we get the suitable size, we compare the toxicity of the moquinone between 2D and 3D cell culture. So this is some of our applications we use just to show that the versatility of the electrospray technology in terms of particle production from millimeter to nanometer to micrometer, and in terms of the material that you can encapsulate using the same setup. We encapsulated hydrophobic material, hydrophilic materials, macromolecules, and even cells. And one of the new students, I think their proposal will be on Friday. I'm co-supervisors. The Nurul will encapsulate bacteria inside the alginate using electrospray as well. So, and for me, one of the most important application of the electrospray is the ability for automation. So we are working on optimizing the dripping using what we call on-demand drop, meaning that you control the number of drops precisely, like perfectly. 1 drop, like you can control like 1,000 drops per second. So if you get a suitable flow rate, or you control the flow rate, you calculate, you will get a suitable size exactly as you control. You can enter the size. I want like 100 micrometer. Then the machine will calculate the number of drops per second, and it will drop that number. That one we already have the prototype, but not yet built a complete setup. But it's already, alhamdulillah, like it's already working up to now. It's up to like 200 micrometer or 100 micrometer. The one that I show one time using the buzzer sound. Okay, so this is what I would like to share today. And this is all from me. Thank you very much. So I hand over back to the MC.
Thank you. Maybe now we come to the question from the floor.
Doctor, I'm not really hearing very well, but it's okay. If there is something, can write in the chat maybe.
Okay. Okay. Maybe question from the floor. Maybe you can ask the floor to ask the questions.
Sorry, doctor.
Can you ask the floor, ask the question?
So, doctor, really your voice is not very clear. Can you type in the chat?
Hello. Hello. Can you hear me?
Not very clear. The sound, doctor. It's very like very dim.
Hello. Can you hear me? Hello.
Okay, doctor. Can you just talk slowly so that I
Okay.
can?
Now thanks for the question.
Sorry, lastly, I forgot to acknowledge some of my students that who heavily work on this technology. Yes, doctor.
Can you hear me? Yes.
Yes.
Okay. Now time for the session. So everyone can ask the question now.
Okay.
So maybe can I ask one question?
Yes, sure, doctor.
Okay, I saw that you already get the pattern, blended already, right?
Can you repeat, doctor?
You already get the pattern blended. Just 1 week ago. So this is about also regarding the spray, right?
The trademark registered, but not yet granted.
Okay, so
But we have 2 other patents related to this one. One granted for the encapsulation of flaxseed oil, and another one great innovation for the large scale setup of the electro spray.
Yes. Okay. Okay. Thank you very much, Dr. Munim.
Okay, thank you.
So I have one question. It's quite echo. Uh, okay.
Yes, I can hear you, Dr. Profile voice clear.
Okay, Dr. Munim. My question is regard to the material that can be what called to produce nano material using the electrospray. Is there any criteria for the material that can be passed through the system?
Basically, any liquid can go through any solution can go through the nozzle, either solution or emulsion can. If it's suspension, of course, it may block the nozzle, but if it's solution, even if it's high viscosity, no problem. But high viscosity solution will give nanofiber. The normal viscosity solution can give you nanoparticles. You can use both organic solvent and hydrophilic or water-based solvent. For hydrophobic solvent, like organic solvent, these chloromethane or ethyl acetate actually is more challenging because they are very fast to evaporate, and they will evaporate in the nozzle itself and block it. You need a very good control of the airflow, the humidity, so a bit challenging in Malaysia. But for liquid, for water-based solutions and emulsions, they are easier to use. You can dissolve the polymers in use the polymers that dissolve in water, like ketosane can be used, alginate, albumin. Some similar polymers can also be used. So you need to find what is the opposite material that can convert it into particle. For example, for alginate, you need to cross-link it with, for example, calcium solution. For ketosane, you may need to cross-link it with TPP, tripolyphosphate. For albumin, you may need to coagulate it with maybe high concentration of salt or organic solvent solution like ethanol 70% or something like this. So this is the one that if you are using a water based solution or emulsion.
Okay. Thank you very much, Dr.
Welcome.
Thank you, Dr. Monia. So for information, Dr. Monia will depart. We will move to USA very soon. So I hope that we still can collaborate with you when you are in USA. So we have working with you. Okay. If no questions, then we'll thank you again, Dr. Monia. And then we will go to the second.
Thank you very much, Prof. I may need to leave after a while. Thank you.
Okay. Secondly, now we go to the second presentation by Dr. Kamal entitled "Optimize the Natural Force Fire Recovery." So we'll talk within 1 hour, including the Q&A. And in addition, I would like to notify the participants. Please, you click the attendance. So Arabic and register. So this will be automatically recorded to the point. Okay, without further ado, invite Dr. Kamal. So now the floor is yours. You can share your slide.
Thank you very much, Dr. Alfi. I share my screen. Can see the screen?
Yeah.
Okay, thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you very much to Dr. Arfi for kind introductions. First of all, I would like to thank Kuliah Pharmacy, especially Head of Research, Dr. Arfi, for the opportunity today to share my research work in the medicinal chemistry. And I'm glad to be here and participating in these forums, and this an excellent opportunity for me to collaborate with you in the future. About 1 hour, we will discuss about my research and potential collaboration in KOP. And our topic is the flavonoids, the natural scaffold-inspired drug discovery. I have been working on this research almost 1 decade, 10 years. And I hope that this can be my sharing mostly of my research during this time. And I open a discussion by hadith narrated by Abu Hurairah. I was put in my PhD thesis related drug discovery, and always I give to the introduction in medicinal chemistry topic to the students. So for my side, it was good meeting to deliver us to find a new drug for the every disease. And the first, we look what is the flavonoids. I think all of us have known the regarding flavonoids are. So basically, flavonoids are a diverse group of phytonutrients. We call the plant chemicals found in almost fruit and vegetables, and it's belong to the natural product chemistry. What is the most outstanding classes of naturally occurring, well-studied compound in the history civilizations, in each responsible for the variety of the pharmacological activity. So it's recognized the potential of the certain regularly occurring structure motif as a template. So for the structure modification to expand the discovery of novel ligands. So this is what we call the prefigured structure. So the natural products play the role in the pharma research in many medicine and drug, either natural product or derivative thereof. And nowadays we have many compounds that are good efficacy. So if you look at the journal, most of the journal, they claim they have a good activity, but they have many lack of the toxicity. The toxicity is important for the leading cause in the attrition in the drug discovery and the development. About 70% of our toxicity-related attrition occur preclinical. Almost these findings are predictive of human toxicity. So natural in synthesis flavonoid-based compounds are considerable. Interests are believed to be non-toxic because they widely distribute in the human diet. So in medicinal chemistry, natural products are robust source of new drugs. So indeed, the FDA revealed that 65% of the 800 more small molecules approved in the market between 1981 until 2019 comprised of natural products. So natural product here is including alternated natural product, biological macromolecules, botanical drugs, natural derivatives, natural product derivatives, structural mimic of natural product, semi-synthetic natural product, analog or synthesis compound based on the natural product pharmacophore. So this shows that how potential our resource in the pharma research. In my research area, cancer and inflammations, a natural product has played a major beneficial role in the cancer chemotherapy associated with the inflammation over 50 years. So both term is of providing established drug and new lead compound for the synthesis optimizations. And this is the the substance for the providing cellular and macromolecular mechanism of action relevant to the cancer and inflammation inhibition. So have a look at the data from the Natural Institute of Health from the year 1940 to 2014. 85 of the 175 small molecules is about the 49% approved in the FDA is the cancer area, were natural product or directly derived from natural products. So it's the significant increase data from the 2014 until now. So a similar trend has also observed in the anti-inflammatory inflammation fields, in which of the 51 of the small molecule approved in the UFSDA, 61% of them is natural products. So this is a sample of the natural product become to the drugs, vincristine and vinblastine. So this is they have the monotherapy in a childhood acute lymphoblastic leukemia. So we look the flavonoids for the model structure. So flavonoid is important hydrocyclic compound of natural product scaffolds because they have the unique structure. Unique structure means that they have a multi-target of receptor possible. So they have the property of metabolite likeness. So this is the natural therapeutic drugs. And also they have a wide class of the natural product. So it can easily modify the structure. And when I began my PhD in 2012, they have two drug approval in Japan. First is the Pranalucas. So Pranalucas is the flavonoid core that has the specific flavon core. First leukotriene antagonist launched in Japan 1995. So in form of the capsule of the oral treatment of bronchial asthma and the allergic rhinitis. And the second is the iguratimoid. Iguratimoid is another flavon spore derived, and is the use for the novel disease modifying anti-rheumatic drugs we call the DMARD, and inhibit the production of the immunoglobulin and inflammatory cytokine. So this is the best sample of the flavon become to the market. And if you look the biodiversity of the flavonoid classes, they divide by two. So first is the major classes. So major classes include the flavones, flavanones, and so on, and also the isoflavones. I think most of you know the biodiversity, and also they have the minor flavonoids. So the narrow objective here, we talk about the minor flavonoid classes, including the hydroxy 2-hydroxy carbones, prenylated carbon, and flavones and aurons. So actually, we synthesis most of the classes flavonoid, and we will discuss further for the next slide. So first, the first step in my research in the exploration. This is the sample I have done the exploration in the Sumatra forest. So my research and teaching explore of some natural product in Indonesia. Started in the 2009 in the Bukit Nang, and 2012 in the Bukit Seligi. So this is my village, my kampung. This I think the Prof Tahir, here Prof Alfi. So the middle of the Sumatra, they have a big forest here. We call the Bukit 30 and Tesonilo. This is Tesonilo. I think this is Tesonilo to 2012. We go to Tesonilo. So this we collect the plant and determine the phytochemistry profiling. This is student try to try to determine the phytochemical profiling in the plant, this pharmacy students, and to teach to students how to explore the potential, how to explore the potential forest in the medicinal chemistry plant. From this, this is a sample, the result explorations. I work with the almarhum Dr. M. Rizal, as the leader to explore forest of the Sumatra. So this is the lignan, and some of steroid we are solid from the Sidia Merah, Piper, Crocatum. So this is the published into the Procedia Scientific. And backward to 2006 and 2007, my YP projects by isolation minor bioactive compound from the Garcinia aqua bring to me to success to xanthan and the tetrapenicoinone. So, and quite interesting. So the ability of the xanthan to interfere with the varietal biology target related with some special molecules, feature such as the presence of the heterocyclic aromatics. And actually, xanthan is belong to flavonoids from the biosynthesis, because from the biosynthesis, xanthan are originated from the cumaret, which flavonoid type C6C3 and 2 acetate unit that indicate that genetic relationship between xanthan and the flavonoids. So I'm work with the Prof. Dr. Darianus in University of Andalas. So we use use the big column to get the minor bioactive compound at the time. And the next exploration we have the isolate pinosandrine from the Gonitanamus macrophyllus, and this is the quite good because we have identified the stereoisomer of the natural pinosandrine from the Gonitanamus by electronic circular discovery (ECD). So this is we hopefully we can be submitted journal, inshallah. If the success I will be share with you. Yeah. Actually, the pinosandrine was approved by FDA, by State Food and Drug Administration of the China for the clinical trial in the patient of the ischemic strokes in 2008, and currently enrolling phase 2 clinical trial by intravenous drips administration of the pinosandrine for the ischemic stroke patient. And we found in this study, we found that the potential to inflammatory for the application for the septic shock. So this is the good data to develop pinosandrine to the septic shock. And in 2009, I start the synthesis, and the beginning, the synthesis, we start the simple compound from the calculus. So I joined the group with the Prof. Dr. Adizambil in the University of Riau. So from here, I have a success synthesis scaffold analog, more than 30 compounds based on the natural product scaffolds. And furthermore, this compound have the potent as the anti-cancer. So this compound are the potent for the cytotoxic again to in vitro, the murine leukemia P3 model with the IC50 about the 5 microgram per ml. So moreover, a micronucleus assay test using the toxicological screening for the potential genocytic compounds was performed for the ability as the cancer effect based on the micronuclei shape to quantify microsomal damage. So this is the we take from the femur bones, and after we treat the mice with our compound. So they have the reduction of the chromosome damage from the we call the initiate the cancer. And moreover, I work on the green synthesis. So as a result, the chalcone has been synthesized in high yield by Claassen-Smith condensation of the substitute acetophenol with the various aromatic dehydes in the presence of the base catalyst at the room temperature by grinding under solvent-free condition. So that time we try to use the very low solvent quantity of the solvents, some of the molecule we success to synthesis without the solvent. We call the solvent-free conditions. Actually, the chalcones are the basic or the initiate or the precursor compound for the other compound. So the one of them is we call the pyroxylene derivative. So this is also we success to synthesis of the pyroxylene derivative. This is a sample of the results. We work on green synthesis for the pyroxylenes, and the pyroxylene was synthesized by improving of the series of the chalcones, react with the hydrogen hydrate at the room temperature under solvent-free also. And the potential anti-inflammatory were evaluated by in vivo, the method we call the pau edema. So the CAR-induced edema in the subplantar region of the right hind paw of the rat, and also affect the inductive peptic ulcer. So the results, its compounds was good inhibit for the anti-inflammatory, and also we not effect on the peptic ulcer of the rat at that time. So we also perform of the docking by web-based method that time in the 2012. And look at the chalcone. Actually, chalcone I have mentioned that have the precursor of the many compound, and especially the chalcone is the precursor for the flavonoid. That is called the building block of the total synthesis of flavonoids, and including the flavones, dihydroxyflavones, flavanones, this is flavones, anthocyanin, isoflavone, auron, and the hydroflavones. This is the how important the chalcones. It lasts 2 decades, a significant rising of interest in the publication and patent related of the chalcones. And we look here, 2 decades and 20 years, they have a significant rising for interest in the publication. So a brief search revealed that the more than 5,000 publications and more than 500 patents published in 1999 to 2008. So this is remarkable double for more than 12,000, near the 13,000 publication, and near the 1,000 patent from the year 2009 to 2019. So this is the how people interesting with the chalcone. They have in general they have the many technique to the synthesis of the chalcone. So the synthesis of chalcone can be easily via synthesis via Claassen-Smith condensations by mixing of benzaldehyde with the active acetobenone under either strong basic or acidic. It's noteworthy to mention that other compound and reaction compound of the calcul, such as Vidal-Crafts, acylation, Novochel condensation, Julia Konsenski, also have been explored with this study. So there are many methods that have to success a story of success to synthesize the calcul. And then this is the quite interesting, the calcul. The synthesis calcul by using the palladium catalysis, and we know that palladium catalysis is the principal Nobel Prize in chemistry in 2010. Akira Suzuki, Negisi, and the Richards F. Heck. So we try to resume what is the important synthesis of the calcul by this technique, and this is the result. We have published in the Journal of the Organic Chemistry, the widely publishing for the technique, the new technique for the synthesis of calcul by palladium catalysis. This is our author, and next we look at how to synthesize calcul first. So in the synthesis, there are many methods and success I have mentioned. The method is depend on the raw material we can use. So we cannot generalize the method. It's the method that will be suitable is depend on the raw material and depend on the moiety in the raw material. So in our finding, after a series of trials, the synthesis of particular calcul was finally success when using the potassium hydroxide 40% with the absolute ethanol. It is sterile in room temperature overnight via Classen-Smith condensation reaction, and get the sum of the variety or the derivative of the calcul. Actually, in the calcul, they have two isomer. We call the cis and trans. This is cis and trans. For my experience, more than 100 calcul I have a synthesis. They have just 2 or 3 compound with the cis isomer. So this is the unique. This will depend on the raw material. It will depend on the method we use. With the calcul in our hands, we also success synthesis of flavonoid, including aurons. This is aurons, prenelated flavones, flavanones, and prenelated flavanones. This is the sample we have done, synthesis and modified the flavanol via calcul as the precursor. This is all the data have been published in the Bioorganic and Medicinal Chemistry letter. How we talk about how to synthesize aurons via calcul. So the success synthesis auron by oxidative cyclization by refluxing of 2 hydroxycarbon with the mercury acetate in the DMSO. This is we have some of the derivatives, and with a bit low yield, it's depend on the raw material and moiety. This is sample, and this is the proposed intramolecular cyclization mechanism of the auron ring formation by mercury acetate. I'm not going to the detail here to the intramolecular cyclization mechanism. And this is the proof by NMR that I have a success synthesis of the auron from the cartoon. And next, we have also synthesis of the flavanol. So flavanol were obtained by modified, we call the IFO, Algar Flynn Oyamada reaction of the chalcone in the 40% aqueous solution of the potassium hydroxide in absolute ethanol, followed by a dropwise addition of the 30% aqueous solution of the peroxide. So the NMR results show the success of the synthesis flavanol. And this is the proposed intramolecular cyclization mechanism of the flavanol via IFO reactions. And important, they have they give the oxidize in the double bond here, and then they can be activated to the nucleotide of this position. Next, we try to another idea. So prenylated group in the flavonoid ring system could be potential increase the lipophilicity properties and bestow to the molecule a strong affinity for the biological membranes. So it should be giving us more active compounds if we apply the prenylated group to our compounds. Here, in the concept, they have a hydrophobic. The cell membrane is belongs to more the hydrophobic group. So this is can give us the more active compounds if we apply to our compounds. And alhamdulillah, we have success. This is sample. We have we concept the proven by our collaboration research with the UPM, with the Prof. Hozira and Dr. Chance. We work was the success to identify the prenylated into the LOX substrate binding sites. This inspires us to apply prenylated into our flavonoid compounds. And here we success to synthesis to insert the prenylated into our group, our compound. Furthermore, we employ to introduce a prenylated into the aromatic system. If first we success into the ether concept. So ether concept is exactly mostly the more easily to insert into the compound, but here we try to employ for the ring system. So this is quite difficult to insert in the ring system. But we have success also by adapted refluxing the compound with the DMA at the 200 degrees Celsius under nitrogen atmosphere to produce intermediate product in the conventional yields. And this is the proposed mechanism we call the signal tropy rearrangement to insert the prenylated into the ring system. So this the method one of the most difficult in the organic synthesis because the yield is extremely low. Here, you can look the yield and need to optimize the reaction condition to obtain proper yield. So this is the mechanism that was the finding by Rainer Louis Claessens in 1912. So we call the terminal orthoclassicity rearrangement, or we call also the KOP rearrangement and para-classicity rearrangement. I mean, I can go deep to explain of the mechanism here. So the success synthesis, they have the movement for the prenylated. This is the proven by NMR. So they move from the O prenylated, we call the here a pro O prenylated to the ring system. To develop the method, we're not working prenulated. So we also use the more long the change. Then we use the 2 chain of the prenulated. We call the generated. So in also we success a synthesis of the generated derivative using a 3.3 signatropic rearrangement, and this is to also give the challenge to us to keep it the stable reaction into the rearrangement. And this bring us to get the new compound with the generated flavonoid and generated 5-member ring of the flavonoid. We call the auron generated, and this in the natural is very we call it very minor flavonoid in the natural. So this is giving us the success of the synthesis of minor flavonoid here. And this is the NMR. We have the proven that compound by NMR, and also with the mass spectrum. Moreover, to challenge the challenge of the natural base compounds is having if the natural base compound have many hydroxyl group on the structure. So the important protecting group in the reaction was focused on this group. For your information, I have been spent 1 year to figure out the suitable protecting reaction in the group. So one of the technique we use the tetrahydroprene ether THP to protecting the group, and we got some of the derivative from here. First is we get the derivative from the calcul, and here look we success to protect by NMR, the proven by hand, and also we unprotected. So we remove the protecting agent. And success here, we also the obvious in the research that have stable in high temperature, because we use the high temperature to work with the auron. So with this protecting, they have a stable in that conditions. Furthermore, we apply palladium coupling reaction to further modification structure of the flavonoid. I have mentioned they have a revolutionary method had brought the founder of the palladium reaction to get a Nobel Prize in 2010. Heck, Negisi and Akirisuzu award in the Nobel Prize in 2010 in the chemistry, developing palladium catalyst cross coupling reactions. And this is our sample that have the success to use the palladium reactions. This is the mechanism reactions for our compound. This is our compound that we propose the mechanism reactions. Then all the compounds all have the synthesis were evaluated for their anti-inflammation effects or the PGE2 and nitric oxide secretion level in the murine macrophage cells stimulated by a combination of the LPS and interferon gamma. You can see here that some of the molecules have the potential inhibitions, and the most active compound is here. This is we look here that prenulated carbones are the most active compound, and it is quite good for us to look that have prenulated, giving us to improve the activity. In the further work, anti-inflammatory effect is most active compound. We go to use some of the activity, and we also do the the docking study from here. And we first elucidate the ligand receptor binding by the computer chemistry. And briefly, PGE2 here is the inflammatory process involves the two enzymes. They are COX-2 and the MPGS1. So then we want to elucidate where is our compound work in the heavy cell as the PGE2. So as a work, we work with the murine. We work with murine cell lines and can easily to find crystal structure of the murine COX-2 in the protein data banks here. But at the time we didn't have get the crystal structure of the MPGS, just available for the human MPGS. We cannot compare the murine and the human MPGS because one is murine and the second is the humans. For look the binding of our compounds, and we know that we use the murine, so we do the homology modeling. We transform from the human MPGS to the murine MPGS homology model. This is the data homology model validation by Raman Chandan plot, and the good quality model would be expected to have over 90% in the most favorable regions. This is the we need to the more further validation of the 3D structure of the murine MPGS. This is the we call the project validation. So project validation is used to ensure that our model is suitable for the further research. And now we have murine and the murine COX-2 and also murine MPGS1. And then we start our docking. Another data, we didn't use the theoretical, just theoretical data. We trying to get the we call the real of the compound, the real of the stereochemistry of the compounds. So we use the crystal structure structure analysis, and from here we know that the stereochemistry of the compound to dock. So this can be give the high accuracy of the docking result. So that by confirmation of the input ligand, so then we have to perform the docking by coordinated of the single crystal assay, and the result is our compound more inhibit to COX-2 murine rather than the MPGS murine MPGS. This is the in silico prediction of the pharmacokinetic properties for the compound. This is we use software to identify the potential ADMET parameter of the compound. To understand whether the compounds could exert similar effect on the human cell, so we opted for the use for the humans U937 cell culture. You can see here there's a very similar activity from the murine in the first research to the human cell, and the compound also subjected to series of the immunomodulatory experimental. So this is to look the compound able to modulate the initial response of the human cell. And I just to share with you how the potential compound. There was one derivative in the Merck, we call the santahumols. The price is very high, with the more than near the 5,000 for the 10 milligrams, and we success to synthesis the new derivative of the santahumol with a good activity. And I have mentioned that the computational approach to translation of the animal receptor model to human models. So this is ongoing research. So this biomedical research to continues to translate from the murine or from the rats to the humans. So this is undergoing the research in our groups. And the other research, this flavonoid also have been determined to the anti-malarial. So we collaborate with the University Gajah Mada, the potential compounds as anti-malarial. For the initial results, we have published in the Scope Journal. We work with the Dr. Jufril Sahri, and also this derivatives compound was published in the Bioorganic Chemistry with the Good Infabetter with the Dr. Siti Munira. After we have a knowledge of several strategies of the flavonoid, we attempt to design the molecules. So this paper we shall continue to our research. And briefly, as we know, we target the COX-2, and the COX-2 was clone of the first clone in the 1992 by DuPont company. That the name inhibitor that time they call DUP 697, and this was important. It's mainly anti-inflammatory assay and didn't have the ulcerogenic effect like the unconventional insights. And at the time, the celecoxib, revacosib was two COX-2 inhibitor that is released in market, and generally based on the UP 697. However, revacosib, valdecoxib were known to increase the risk of the heart attack. So that the drug withdraw from the market in September 2004 and 2005. And what we do, we collect the structure of the COXIB. We collect the structure of the COXIB drugs. We know that the COXIB drug have a good efficacy. That means there's no doubt efficacy of the drug, and give the low side effect for the ulcerogenic. So this is we try to collect the drugs, and what we do after we collect the drug, we cutting off 5 membranes of the COXIB drugs. We replace with the common ring. So this is the theoretical drug that maybe could be potential with the similarly active with the COXIB, but with the low toxicity. So the question is how synthesis this target drug. So first we do the retrosynthesis analysis. The analysis, this is the target drug, the theoretical drug. We trying to look the retrosynthesis analysis to find a raw material to initiate synthesis. And alhamdulillah, from here we get one drug from here, one molecule, and this molecule could be potential active similar with the COXIB drug, but give the low toxicity, because we know that the flavonoid give the low toxicity because the natural base compound I have mentioned in the first slide. This is some of the technique to get the molecules. We call the bromization mechanism of the flavones, and also we use the Suzuki coupling reaction for the compounds. The compound. This is our proposed the mechanism reactions. And 4 steps from here, from the chalcone and flavons, bromization, and the Suzuki coupling reaction. The last to give our target drug, our target molecules. We confirm the structure by crystal structure and by crystal structure and NMR also here. And the structure is quite correct from the stereochemistry. So this is bring us to knowledge how to synthesis more derivative from this compound. Moreover, this compound we success to identify the potential anti-inflammatory effects. They have a good selectivity of the anti-inflammatory effect from the success to inhibit COX-2 rather than COX-1. And we generate the pharmacophore from here to get the new what is important pharmacophore in the molecules. This is the molecular docking we have success to study for the inhibiting of the COX-2. And also we look more deeply for the binding with the molecular dynamic simulations here. So in the molecular dynamic simulations, they have good binding and stable for that time for the several time of our analysis. That means the compound is the have very good for the inhibition into the active site. Sorry. Sorry, I have a technical problem here. Sorry, I have a technical problem here. I continue. So this is the summarize of the molecular dynamic simulation snapshot of the overall interaction into the binding site. And moreover, we also didn't work with the murine. We also look with the human cell. So here, and also they have good inhibition with the humans. So that means that they have they have a correlate with the murine and the human for this compound. And also they have the selectivity, good selectivity. So this is just I'm share my research. We use also our blood to this work. And some of the compound we also try to look the mRNA level and by PCR. So this is the have good inhibition of the COX-2 mRNA by the selective of the COX. And from here we try to look what is exactly pathway to the compounds, and we goes to use the reverse docking. So reverse docking means that compound we try to dock to some of the several of the enzymes, and we compare with the original ligand. And here, our compound more suitable with the P38 alpha from the particular pathway. And this is the molecular dynamics information for the P38 pathway, particular enzyme into the active site, and they have a good good binding into the active sites, and that's meaning that they have the good inhibition property into the active site of the P38 mechanism. Then we goes to optimize the compounds. We use the computational approach by essential rule. We go to 3D CubeSAR, 2D CubeSAR for that compound, and we get the some of the idea how to improve the compound. So they have a 3 strategy actually, ABC strategy to improve the compounds, and most of the strategy we have the success synthesis compounds, and from here mostly 60-70 compounds have success synthesized by several of the methods. And I hopefully for next we will publish soon for these compounds. First, we have looked try to inhibit the inflammation in the COX pathway. Now we try to look the other pathway. We call the LOX pathway, leukotriene pathway. So in this mirror, we focus on here 5 LOX, lipoxygenase, and the FLAP. So actually, FLAP is the activating protein for the LOX. So LOX is not the work with the alone, but they need to be activated by another enzyme we call the FLAP. So some of the molecule, this target, still in the preclinical trial, not become drug anymore. So the sample is the N103. The first become, not yet, become the first FLAP inhibitor. They have in the phase 2 clinical trial, phase 3 or phase 2 clinical trial by Amira Pharmaceutical. This is our interesting for the concept. So from this concept, unexpected results, we get the other flavonoid from here. We have success screening from the Arsenex database from the 200,000 compounds from here, and we do the sum of the filter screening here and got from the 10 compounds, and some 3 of compounds from here they have the there is the calculus. That is the interesting for us. Now we try to synthesis of this carbon. And the other work we also do to relate the anti-inflammatory and the cancer. So we got the one of the concept from here. We call the autophagy. The autophagy, I think, it's a Nobel Prize in 2000 in medicine in 2018 or 2017. This is important for our research now. And one of our flavonoid, they have a good inhibit an autophagy, ULK1. So this can be good data to get new target of the cancer via inflammatory. This is my student PhD work, Sister Novarisa, with the Prof. Tahir and Dr. Ina, Prof. Tahir, and we collaborate with the Prof. Andreas from the Austria, and Dr. Taimur Luhat Mongkol from the Thailand, and also from the Magna Cancer in the UPM. We still ongoing of the research, and we will publish soon for the research for the initiate data, inshallah. And this is the two of the development of my research. They have the antimalarial from this flavonoid, a xanthine oxidase, LOX inhibitor, and FLAP inhibitor. This is my another analogs from the natural compounds. This is the curcumin. And we have several published for these compounds. Okay. The conclusion, flavonoid are important source for the potential treatment of the multifactorial disease. However, effort toward the development of the flavone-based therapeutic agent are still needed. So the appearance of new catalysts and new chemical transformation is expected to provide a new way for the synthesis of the unexplored flavones, leading to discover of the flavones with the new properties and biological activity. This is my collaborations. Thank you very much. I give back to Dr. Arfi. Thank you very much.
So, Dr. Kamal, very nice, very interesting session. We have a very huge data presenting. Maybe any questions from the floor? But then maybe I have one question, Dr. Kamal. Hello.
Sorry, doctor, I cannot hear well
Hello.
from here.
Can you hear me? Can you hear? Dr. Kamal?
Yes,
Boleh, dengar?
a bit,
Dengar?
not very clear.
Okay. Can you read the chatting box?
Okay. Okay, from the Dr. Sam, I have two questions. First, what is software can use for docking? Second, what are the receptor or a protein that we target docking? Any other example another than COX? Okay, thank you very much, Dr. Sam. Actually, the docking, I use the not free software. I use the Discovery Studio software. I'm collaborate with the UKM because the UKM they have the license, and also I'm collaborate with the Dr. Zalika. We try to find the free software for this project also. We try to develop to more easily use for the software. And the second, the receptor of protein. The protein we target first is the COX. From my beginning of my research, I target the COX from the 2012 until now. I try to COX, but during that times, we also discover some of the inflammatory pathway, because inflammatory in cancer pathway is meaning a lot of the protein involved there. Some of protein is new. If a protein is new, it's very difficult to buy because it's very high price, very expensive, and some of the like the flaps, we try to develop the flap in this world. In the rules, I try to collaborate with the Amira Pharmaceutical, but they say they have very confidential that particular protein cannot collaborate because the new protein. Then they say that there is the working on that, no drug from there, so they didn't prefer for the collaboration. So we also we try to look another protein. So we work with the COX. We work with the LOX also for the inflammation. We work for the autophagy, mTOR, ULK1, and MEP kinase. So MEP kinase also we work the P38 MEP kinase. We work with the JNK and we will go for the ERK. Not all mechanics, because mechanics I think have found 300 mechanics more. So we just look our specific target from there. Thank you very much, Susan.
Can you read the chat box? I send the question.
The other question is kindly elaborate the toxicity of the synthesis compound produced from the UO. The toxicity we try to divide by two method. First is the in silico study. So in our software we can determine the toxicity at map and toxicity, and the toxicity we can determine 15 kind of the cytotoxic toxicity. So this can be used for our design. This can the molecule can be figured out to the next level, or we just stop for the here, and we try to the other compound. And the second, we use the experimental. Of course, we use the simple experimental by toxicity by normal cell to normal cell and other toxicity. So basically, we use the normal cell toxicity. Sorry, Dr. Alfie, I cannot hear you.
Okay. Hello.
Yeah.
Dr. Kumar.
Yeah. Dr. Alfie.
Okay. Sorry. The technical error in. So president, with that, this is we already come to the end of these sessions. For the next KOP Tarik session, for information, we will have hybrid mode, meaning that we will provide food, the tea, high tea, for all participants. So now actually we already have also the tea in the kuliah meeting room, so you can go downstairs and collect the Tarik and also some small cake. So with that, let we end our session with the tasbih kafarah and surat Al-As. Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much. Waalaikumsalam warahmatullahi wabarakatuh.